Everyone, and welcome to Miss Estruch Biology, and in this video I'm going through everything you need to know for AQA GCSE Biology in this one entire video, so you've got your revision sorted. Now if you are pushed for time, don't forget you can watch this at ×2 speed to get through it quicker, but to make sure you get the most value out of this video, have a go at pausing, writing down key terms and testing yourself as you go. And if you really want to boost your grade, don't forget to check out my GCSE notes, which cover all of the theory, mark scheme specific, with examiner tips, key terms, key definition, topic summaries, and question and answers at the end of each topic too. Best of luck in your GCSE exams, everyone, especially WCHS students. So topic 2 for GCSE Biology. First of all, we're going to go through this introduction of what's coming up in this video, but I do want to point out that at the end of this video, there are also 10 questions and the answers to test have you understood all of the information. So don't miss those questions at the end. But introduction then. This topic on organisation is looking at how the body is made up of complex systems and how they all work together to keep us alive and healthy. So you learn about the digestive system, respiratory system, circulatory system, and then also the transport systems in plants. And there's a little bit as well about what could make these systems go wrong and things like coronary heart disease, how you could overcome those. So this term organisation is referring to all of the different structures within an organism and how those increasingly complex structures work together to create a final organism. You need to know about cells, tissues, organs, organ systems and organisms. Cells was all of topic 1, which if you haven't watched my video on topic 1, I'll link it up here. So cells are the smallest of the structures. They're the basic building blocks of life. Then we get tissues, which could be made up of thousands or even millions of cells, and they're groups of similar cells that work together to carry out a specific function, such as a muscle tissue. The next structure up in terms of size is organs, and organs are made up of different tissues working together to perform a particular function, for example, the stomach. And then organ systems are lots of organs grouped together, which work together to perform a function, like the digestive system, which we'll be covering. And then the largest is an entire organism, which means a living thing, and organisms are made up of different organ systems working together, and an organism could be a human, for example. So then if we move on to some examples of each of these structures, you need to know about tissues in animals, organs in animals, and organ systems in animals. And the first organ system is the human digestive system. Now, it does say in the spec that you need to know the digestive system in the level of detail from Key Stage 3, but I'm guessing you might not remember that, so I'm going to go through that information so you know exactly what you need to know for your GCSE exam. So the first thing is, what is the function? The digestive system is made up of lots of different organs. Some of these aren't the digestive system. We can see the lungs, but we'll go through all of the ones which you need to know. And the function is it breaks down your food from being large and insoluble molecules into smaller, soluble molecules which can be absorbed into the bloodstream and then transported around the body. Any food that isn't absorbed after digestion then forms the feces and is removed as waste. So the structures of the digestive system, or the organs that you need to know, are all the ones listed here. So you might want to screenshot, print it and have it in your notes, or you can copy out the table or turn it into flashcards. So we've got the mouth, that is where we have mechanical digestion, which means chewing to break apart the large molecules. You also have some chemical digestion where we have enzymes, which we'll talk about later in the video, such as amylase, which help to chemically break down some of the larger molecules. The salivary glands, which we can see just here next to the mouth, those are producing and releasing saliva, which contains the enzymes that do some of the chemical digestion. Then the food is swallowed and it goes down the esophagus, which is this tube here. Ignore the spelling in this picture. This is the UK spelling of esophagus, and that's just a tube that connects the mouth to the stomach. Then we get to the stomach, and this is where the food is mixed or churned up, and it's mixed with acid, hydrochloric acid, but acid is sufficient, and that is to partially help to break down the food, but also it's to kill any microbes like bacteria that might have been in your food to protect you from getting an infection and getting ill. You also have some enzymes which are there to digest proteins. Once that has happened, the food then passes into the top of the small intestines, and that top part of the small intestines is known as the duodenum. The next bit is the jejunum here, but you don't need to know that. You just need to know about the duodenum. That is where the food passes into. And at that point, we have the gallbladder shown here in green, and the pancreas, you can just about see it here behind the stomach shown in yellow. Those both connect to the duodenum and release liquids into it. Now, you need to be more specific than just saying liquids. You need to say what those contain. So the pancreas is going to be releasing or secreting liquids that contain enzymes. And we'll cover these 3 enzymes later on in this video. Now, this bit here about the bicarbonate, ignore that, because what you actually need to know is down here linked to the liver and the gallbladder. And that is the liquid known as bile, which is actually this like bright green sort of colour. And the liver is where bile is produced. The gallbladder is where it is then stored, and then it's released into the duodenum. And we'll be learning about what bile is, but in brief, it emulsifies fats, which means splitting it up into smaller droplets. And also it is what neutralizes the stomach acid. So when the contents of your stomach then moves into the duodenum, it neutralizes that contents so that you don't get damage to the small intestines. Because your stomach can withstand the hydrochloric acid because it's got a special layer, but the small intestines doesn't have that, so it would be damaged if we didn't neutralize that stomach acid as it travels in with the bile. Then the small intestines, the rest of the small intestines, this is the main site of digestion with enzymes, and it's also where the digested food gets absorbed. One key adaptation of the small intestines is the inside of this is highly folded, and that is creating these structures known as villi, which increase the surface area for the absorption of the digested food. Once most of that content is then absorbed, it will then transport into the colon or large intestines. And this is where water is absorbed. So that's the main role of the large intestines. And then anything that hasn't been absorbed then gets stored in the rectum, and then eventually it is released from the body as feces through the anus. So we talked about enzymes quite a bit there, but we haven't said what an enzyme is. So here we can see an image of an enzyme. Enzymes are biological catalysts, and that means they speed up chemical reactions. And it's known as biological because it's happening inside of an organism. They are essential for metabolic processes, which, if you aren't sure on the definition of metabolism, that means the sum of all the chemical reactions in a cell or organism. So in other words, it's all the chemical reactions that happen in your body. So enzymes are speeding up all of the chemical reactions that happen in your body. And they are able to do this because of this unique shaped part in them known as the active site. And this is where substrates can bind if they are complementary in shape, meaning opposite in shape. And there is one enzyme to catalyze every different reaction, because there are lots of different enzymes which all have a different shape active site, which is specific and only complementary to one substrate. So let's have a look at how the binding of the substrate to the complementary shaped active site speeds up these chemical reactions. So that active site is the key point on an enzyme. And enzymes are large proteins which are folded up to make these unique 3D shapes. And that's how we get this unique shaped active site. And as I said, that active site will only be complementary in shape to one substrate. And when we say complementary, we mean opposite. So they fit together like pieces in a jigsaw. And that is what is going to happen to enable the enzyme to speed up the rate of reaction. So when that substrate binds, that is then going to put some tension on the substrate, so it will help to break it apart using less energy, and therefore it will be a quicker reaction. Now, this is actually explained in more detail through this model known as the lock and key model, or the lock and key theory. And this is explaining how enzymes speed up or catalyze reactions. So first of all, it is suggesting that the enzyme, in particular the active site, is like a lock, and the substrate is like a key. And only if that key is perfectly complementary in shape to the lock, which in this case is the substrate, being complementary in shape to the active site, can it fit in? When it does fit in, it is known as an enzyme substrate complex. So in terms of the lock and key theory, this is when the key would be inside of the lock. And this term enzyme substrate complex is almost always worth a mark in exam questions linked to enzymes. So write this down, put it on a flashcard, make sure you know that key term, and any enzyme question you get, where possible, try and use it. And if you use it correctly, then it'll be worth a mark. And that's because when this enzyme substrate complex forms, that is what is catalyzing the reaction, because it's helping to break apart the substrate. Or if it's a reaction where you're joining two molecules together, it's holding them in place and making it easier for the bonds to form. So this is allowing the enzyme to carry out its function. Once the substrate has been broken apart or joined together, depending what type of reaction it is, we then get an enzyme product complex, and it's slightly different shape to the substrate, so it then is released. Once the products are released from the active site, the enzyme can then be reused to catalyze that reaction again, assuming you still have more substrate to react with. So the enzyme is unchanged, it doesn't get used up, and it's just there to catalyze the reactions. Now, enzymes are affected by certain conditions, and you need to know how temperature and pH affect enzyme activity. So first of all, let's have a look at temperature, starting with what happens if the temperature gets too hot. Enzymes have what is known as an optimum temperature, and the word optimum means the best temperature for the enzyme. So basically the temperature it works fastest at is the optimum. And for most enzymes, the optimum is around 37 degrees C, because that is body temperature, and enzymes are catalyzing the reactions inside of the body. So that is typically your optimum. Temperatures above the optimum, so higher than 37, will start to denature the enzyme. And what denature means is it changes the shape of the enzyme. And if you change the shape of the enzyme, the active site will change shape. And once the active site has changed shape, that is what we mean by an enzyme denaturing. Now, this means that the reaction won't be catalyzed anymore, because if the enzyme's active site changes shape, the substrate can no longer bind because they're not complementary in shape, and you don't get enzyme substrate complexes forming. Therefore, the rate of reaction will decrease, and if all enzymes denature, then the rate of reaction will stop. Now, if it's too cold, enzymes do not denature. What happens at the colder temperatures is the enzyme and also the substrate won't be moving very quickly, because if it's cold, the molecules will have less kinetic energy, which is basically movement energy. And if your enzyme and substrate are moving around slowly inside of your body or inside of your cells, then they're less likely to come into contact with each other and collide, so that the substrate can collide into the complementary shaped active site. So that means we get fewer enzyme substrate complexes, and again, the rate of reaction will therefore be lower. So two key things to make sure you do not make a mistake with. Number one is, if it is cold, enzymes do not denature. It's just you're less likely to have successful collisions because the molecules are moving less. Number two is, enzymes are not living things, they're protein molecules. So enzymes cannot be killed. If an enzyme isn't working, you have to say they've been denatured. So next then is looking at the effect of pH. Enzymes work best again at a particular pH, which is known as the optimum pH. But in this case, enzymes have different optimal pHs because different parts of your body are at different pHs. So for example, in the stomach, the pH in your stomach is around pH 1 or 2. It's very, very acidic. So the enzymes that you find in the stomach, like protease, which digests proteins, they have optimum pHs of 1 or 2. But then the enzymes that you find in the small intestines is about pH 8. So they have an optimum pH that they work at of 8. So whatever the optimum is, if the pH is either too high, meaning it's too alkaline, or too low, meaning it's too acidic, that causes the enzyme to denature as well. So it also causes the enzyme to change shape, changing the shape of the active site, preventing enzyme substrate complexes, and therefore reducing the rate of reaction. So just to summarise some of those key points, we said our definition of metabolism, that is the sum of all chemical reactions in a cell or body. And metabolism is reliant on enzymes. Now, some examples are breaking down large molecules, which we're going to have a look at in digestion. Some of them, though, involved helping to build larger molecules. So one of the Year 11 topics is protein synthesis. And then we've got energy transfers. So when you learn about respiration and photosynthesis, they also involve enzymes. So all of these are enzyme controlled reactions, and all of these make up metabolism. So let's have a look then specifically at enzymes in digestion. And these are enzymes responsible for breaking down molecules. Specifically, they're breaking down the large insoluble food molecules into smaller soluble molecules. And those smaller molecules are then absorbed into the bloodstream and transported around the body. So here are the enzymes you need to know. We're going to go through all of them in more detail, but again, just in case you wanted to screenshot and have it all on one page, you can add it to your notes. Here's your screenshots. But I do just want to point out for the amylase one, if you are taking a screenshot, can you add next to it carbohydrates? And amylase is an example of a carbohydrate. So here's our summary, but let's go through each one. Number one are the carbohydrate enzymes, and these digest carbohydrates, and they break down or digest those carbohydrates into smaller soluble molecules, which are simple sugars. So amylase is an enzyme, which is an example of a type of carbohydrates. So the site of production, meaning where are these enzymes made? So some are made in the salivary glands, which then get secreted into the mouth to work there. Some are produced in the pancreas, which then gets secreted into the duodenum, that top part of the small intestines. And some of them are made in the small intestines and then are used in the small intestines as well. So here's a very simple word equation. An example of a carbohydrate is starch. Starch is digested by amylase, and it's digested or broken down into the simple sugar maltose. So one little tip to try and help you to remember new terms in biology is any word that you see in biology that ends in ASE means it's an enzyme. So carbohydrates, that literally means it's an enzyme that breaks down carbohydrates. So amylase is an example of a carbohydrate enzyme, and it ends in ASE. The second digestive enzyme is protease, again ending in ASE, so we know it's an enzyme, and it digests proteins. So the name of that enzyme is implying what they are digesting. Proteins are polymers, and they are digested into the smaller monomers that make them up, which are amino acids. The places that protease are made are the stomach. So we have some digestion of proteins happening in the stomach. The pancreas, which will release the enzyme protease into the duodenum, which is that top part of the small intestines. So there'll be some further digestion of proteins in the small intestines. And then the small intestines themselves also produce proteases for further digestion there. So here's a simple word equation. Protein, which is our large insoluble food molecule, that is digested into its monomers, the amino acids, which are smaller soluble molecules, and that is done using the enzyme protease. The last one you need to know is lipase, ASE at the end, so know it's an enzyme, lip at the start because it's lipids. Lipids is the name for things like fats and oils. And lipids are digested into two molecules. We get glycerol and fatty acids. So a lipid is split into those two different molecules. Lipase is produced in the pancreas, and it's secreted from there into the duodenum, which is part of the small intestines. And also you get even more made in the small intestines. So the only place lipids are digested is the small intestines. And here we have a simple word equation. The lipids are broken down into glycerol and fatty acids with the help of the enzyme lipase, sometimes pronounced lipase. But you can say either or, just make sure you spell it correctly. Now, there's a little bit more to lipid digestion that you need to know about, and that is the role of bile, which we briefly talked about when we looked at the overview of the digestive system. Bile is not an enzyme, but it does help with the digestion of lipids. So bile is this green liquid that is made in your liver. It then gets stored in the gallbladder, and from there it's released into the duodenum, which is the top part of the small intestines. And bile does two things. Number one, bile is really alkaline. So that means as you add bile to the top of your small intestines, and you have the stomach acid and the contents of the stomach pouring in at that point as well, that strong acid from the stomach will mix with a strong alkaline, which is bile, and it neutralizes that hydrochloric acid, that stomach acid, and it actually makes it slightly alkaline. We get about pH 8. You don't need to know that though. And that is now the optimum pH for the enzymes in the small intestine, such as lipase. So that's one of the jobs. It neutralizes the stomach acid to get the optimum pH for enzymes to work at. The second job is bile emulsifies fats. And what that means, if I just do a quick diagram, if we imagine a fat droplet, whether it's a droplet of oil or it could be a droplet of a solid fat, you've got one large droplet, one large sphere. Bile is able to break that one droplet into several smaller droplets. And that's what emulsification means. It's when you are separating that large lipid droplet into lots of smaller droplets. And the reason this is an advantage is if you've got lots of small droplets, that now means you've got a greater surface area for the enzyme lipase to act on. So let's imagine here we could maybe only fit one enzyme on, but now we'd be able to fit lots of the enzyme lipase on. We've got a greater surface area, so that's going to speed up the rate at which lipase can digest the lipids. So let's have a look at the products of digestion. We said from digesting carbohydrates, you get simple sugars. And one of those simple sugars is glucose. Glucose can be used in your body to build new, larger carbohydrates, or a lot of it is used immediately in respiration to release energy. We also had amino acids, which were produced from digesting proteins, and those are used to synthesize, which means to create, new proteins in your body. And your body is actually about 50% proteins if you were to remove all the water. And that's because things like enzymes, muscle tissue, antibodies, which you'll learn about later, these are all proteins, so amino acids are essential. And then finally, the glycerol and fatty acids. Once those have been absorbed into the bloodstream, they can then be converted back into lipids, which are used for things like making your cell membranes and also energy storage. So we then move on to the next organ system, which is the circulatory system. And the main organ that you learn about in the circulatory system is the heart. The heart is a muscular organ, and it pumps blood around the body as part of a double circulatory system. We're going to come on to that concept in a few slides' time. The main function then of the circulatory system is to transport oxygen and nutrients around the body. And when we say nutrients, that's things like we just saw in the products of digestion. So glucose, amino acids, fatty acids, glycerol, so that cells have everything they need to be able to release energy or to create new substances. But we also need to remove waste products so it doesn't become toxic in the body. So things like carbon dioxide need to be transported around the blood to get to the lungs so it can be exhaled. So here is the route that blood takes. But before we go through the route, let's just have a look at the key structures. The heart is made up of 4 chambers, which basically means rooms, so 4 spaces. The top 2 are the atria, or atrium for singular. So we've got the right atrium and the left atrium. And just as a note, when you write it down, you have to imagine that if you were to pick up that picture and hold it against your chest, that is what we're considering the left and the right, not how it looks to you looking down at it. That's why that side says the right. The bottom 2 chambers, which are larger, those are known as the ventricles. So here is our right ventricle. And then on this side, we have the left ventricle. So those are the 4 chambers of the heart. You also need to know the 4 blood vessels that are attached to those 4 chambers. So the right atrium, the blood vessel attached to it is known as the vena cava. Now this is Latin. Vena means vein, cava means body. So the vena cava is the blood vessel carrying blood from the rest of the body into the right atrium. Coming out of the right ventricle, we have the pulmonary artery. Wherever you see this word pulmonary in biology, that means it's attached to the lungs. An artery is a blood vessel that carries blood away from the heart. So the pulmonary artery is a blood vessel carrying blood away from the heart and taking it to the lungs. Then we have the left atrium. Going into the left atrium is the pulmonary vein. So we know this must have come from the lungs because it's got the word pulmonary in it. And vein, those are blood vessels that are transporting blood back into the heart. I always say think it's got the word in it. It's taking the blood back into the heart. And then lastly, we have the aorta. This is the blood vessel. It's an artery carrying blood away from the heart and to the rest of the body. So those are the 4 key blood vessels that you need to know, the 4 chambers. A few other things just to point out, which are visible in this picture, are we can see we've got what's labelled heart valves. So this flap here and here, that is a valve. We've got them here, these 2 flaps, here as well and here. Valves we're going to talk about a bit later, but just in brief, they can close to make sure blood flows in the correct direction and only in one direction around the heart. The last thing I just want to point out is the thickness of the muscle. So look at how thick the muscle is in the left ventricle wall compared to how thick it is in the right ventricle wall. And this is because the right ventricle is pumping blood, we said up here, to the lungs. And the lungs are right next to the heart. So you don't need to have muscle contracting with that much force to pump the blood to the lungs. So this enables us to pump the blood at a lower speed, lower pressure, so that you don't damage any of the blood vessels in the lungs, and also so the blood flows slower, so there's more time for gas exchange. But the blood that is in the left ventricle and is then pumped out of the aorta, that's going to be transported to the rest of the body. So it's going to have to go from the heart all the way down to the cells right at the tip of your feet. And to do that, it needs to be pumped out at high force and high speed. So to be able to pump it at high force, you need to have thicker muscle so it can contract more powerfully. So that's why the left ventricle has thicker muscles. It's so that it can contract with more force and therefore pump the blood out at higher pressure. Final thing I just want to point out is these arrows are showing the routes that blood takes. We'll talk through it here in this diagram or flow diagram here. But you also see it's color coded. So you've got blue arrows versus red arrows. The blue arrows mean that this is deoxygenated blood. And that's because it's blood that has been delivered from the body where the cells have used up all the oxygen in respiration, and it now needs to go back to the heart to be pumped to the lungs to become oxygenated again. And because the pulmonary vein is delivering blood into the heart from the lungs, got that word pulmonary, that means if the blood's just come from the lungs, it's going to be oxygen rich, or in other words, oxygenated blood. And that's what the red arrows are representing. It's oxygenated. Now this here talks us through the route of the blood through the heart. But this is imagining if we were looking at the journey of a single red blood cell, the route it would take all the way around. Because in reality, as we can see here, those 2 arrows both pointing in, blood is going to enter from the pulmonary vein and the vena cava at the same time. So blood enters both sides at the same time and is pumped out of both sides at the same time. But if you had to do a long answer question describing the route, it often says a start point and you have to describe the whole journey. So if we start at the vena cava, where we can see here, that takes blood into the right atrium. Then from the right atrium, it's pumped into the right ventricle. Then we can see from the right ventricle, the blood is pumped up and out of the pulmonary artery. That is then taking the blood to the lungs. From the lungs, it gets oxygenated, and then the blood re-enters through the pulmonary vein into the left atrium. From the left atrium, it's then pumped into the left ventricle. The left ventricle contracts with the most force to create high blood pressure, pumps the blood up and out of the aorta, and that then takes the blood to all of the body tissues, and then we go right back to the start again, the vena cava. So that is the structure of the heart, some of the details about it, and also the route that blood takes through the heart. So this information here is actually summarising some of the previous facts I'd already annotated on. So just to summarise those, the right side of the heart shown in blue is handling deoxygenated blood. The left side of the heart has oxygenated blood. Gas exchange, we're going to talk about in more detail when we get onto the lungs. That is happening in the lungs. And the left ventricle has the thickest muscular wall because it has to contract with more force to pump blood out of the heart at higher pressure so it can reach all of the body cells. So next then, let's have a look at the heart valves. And I've actually already annotated this onto the picture and highlighted them. And we briefly mentioned it, but we'll go through this in more detail. So heart valves are basically small flaps, and when they close together, it stops the blood going backwards. So they're a bit like doors, making sure that the blood travels in one direction through the heart. And we have them between the atria and the ventricles on both sides, and we have them between the ventricles and the arteries, so the pulmonary artery and the aorta. So the whole point is valves prevent the backflow of blood. That's the key phrase that is used in mark schemes. And the reason this is important is it makes sure that the blood flows continuously in one direction. And that's important for the efficient transport of oxygenated blood to your respiring cells. So there's like your mark scheme answer for describing and explaining the role of heart valves. Next then is the coronary arteries. These are actually arteries on the outside of the heart. So far, we've just been describing the inside of the heart, or the chambers inside. But the coronary arteries are blood vessels that run on the outside of that heart muscle. And the coronary arteries supply the heart muscle with oxygen and glucose, which are needed by the heart muscle so they can respire and release energy, because the heart is constantly contracting and relaxing. But for a muscle to contract and relax, it needs energy. And that energy comes from respiration, and respiration needs glucose and oxygen. So if you get a blockage in one of your coronary arteries, which is what this image here is zooming in and representing, these fatty deposits, blocking it so that blood can't flow through anymore, that can result in a heart attack because it would mean your heart muscle isn't getting oxygen and glucose. That means it can't respire. That means the muscles can't contract, and that's what a heart attack is. It's when parts of or all of your heart muscle isn't contracting anymore. So a double circulatory system then. At the start of this section, I said that the circulatory system is known as a double circulatory system. So what that means is there are two circuits that the blood travels through, and therefore the heart has blood travelling through it twice. So the first circuit we can see here is the pulmonary circuit. And if you remember, we said pulmonary means it's attached to the lungs. So this is the circuit where the blood goes from the heart to the lungs, back into the heart. So that's just one circuit that the blood takes. The second circuit is the one that goes to the body, known as the systemic circuit, but it'd be fine to just call it the body circuit, or to say it's the circuit where the blood is transported around the rest of the body and then it goes back into the heart. So the advantage of this is it means that we can transport the deoxygenated blood to the lungs to become oxygenated, and it means then to the rest of the body cells we can transport the oxygenated blood, which is a really efficient way to transport oxygen. So we have really oxygen-rich blood rather than mixing the two together, and that would just dilute the amount of oxygen you actually have present. It's also really helpful because it means that the blood can be pumped to the lungs at a lower pressure than the blood that is pumped to the rest of the body. And that's good because we want the blood to go to the lungs at lower pressure so that the blood flows through the capillaries in the lungs slower. And that means there's more time for oxygen to diffuse into the blood. And it also means if it's going at a lower pressure, it's less likely to cause damage to those blood vessels in the lungs. Another advantage is it means that the blood can be pumped out of that left ventricle through the aorta to the rest of the body at higher pressure. And that means that the oxygenated blood is going to be able to reach all of those respiring cells in the body. Now, sometimes you have issues that go wrong in the heart. And inside of your heart, you naturally have a group of specialised cells, or in other words, a tissue, because a group of specialised cells performing a function, that is a tissue, known as the pacemaker. And you find them in the right atrium of the heart, one of those top chambers in the heart. And the role of these pacemakers are that they cause this release of electrical activity. It's this wave of electricity goes across the heart, and that is what causes the heart muscle to contract. And the frequency, so how quickly the pacemaker cells release this wave of electrical impulse or wave of electricity across the heart, is what determines how quickly your heart beats. So in other words, the pacemakers are setting the pace of your heartbeat. So they are dictating how quickly your heart beats. And this is really important because first of all, it ensures you have a regular heartbeat, but also it's coordinated to your needs. So if you are resting, the pacemaker will release that wave of electrical activity slower. If you are doing exercise, it will release that wave of electricity faster. And in that way, it can control how quickly your heart beats. In some cases, though, people's natural pacemaker cells stop working. And as a result, you might get an irregular heart rhythm. And that could be dangerous because it means you're not then actually pumping blood around the body at set frequencies that you need for respiration. So when this happens, you can use an artificial pacemaker. So an artificial pacemaker, this is going to be implanted just under the skin, usually around your neck, at your collarbone, and then you just have a little cable that goes through blood vessels to reach the heart. And the artificial pacemaker will release that wave of electricity instead, and that will now be what is controlling your heart rate. So that would be the solution or the treatment if your natural pacemaker cells weren't working correctly anymore. So you've just got a little summary box there on the difference between your natural pacemaker cells, and because it is a group of cells, this is an example of a tissue, versus an artificial pacemaker. Right, we then move on to our next organ system, which is the respiratory system containing the lungs. So the human lungs are vital organs in the respiratory system, and this is where we have gas exchange happening. So oxygen is going to diffuse from the lungs into the blood, and carbon dioxide is going to diffuse from the blood back into the lungs to be exhaled. Now, this process is known as gas exchange because you are exchanging gases. Gas from the air is exchanging the oxygen into the blood, and the gas carbon dioxide goes from the blood back out again from your lungs and exhaled. Specifically, the gas exchange happens at a structure known as the alveoli. So we're going to have a look at the structures of the lungs in more detail. One thing though I just want to point out before we do this, a really common mistake that is made is that because this is known as the respiratory system, people think that inhaling and exhaling is respiration sometimes, and that is not true. Respiration is a chemical reaction that releases energy. What happens in the lungs is gas exchange, and breathing is what ventilation is. And once you've breathed, you can then have gas exchange. Okay, so the key structures, we'll start with the trachea, which is this pipe we can see here. So this is a tube, also known as the windpipe, which has cartilage rings. So we can see these rings here. Cartilage is a slightly flexible but also hard material. So if you feel your nose or your ears, you don't actually have bone in your nose or your ears, but they have a solid sort of structure to them, and that's cartilage. So your nose shape is cartilage, your ear shape is from the cartilage. And you also have rings, C-shaped rings of cartilage in your trachea. And that's to make sure that your windpipe or your trachea is permanently open and that the tube doesn't get sticky and stick together. So this is the tube that connects to your mouth and your nose, and it carries the air as you inhale to your lungs. There's also tiny hairs that line the inside of the trachea known as cilia. And these hair-like structures, they move back and forth in this sort of sweeping motion. And what then happens is the mucus that also gets produced by cells lining the trachea. And the whole point of mucus is it's this thick, sticky substance. So when you inhale, any dust that you might have inhaled or any microbes that you might have inhaled, they should get stuck to that thick, sticky mucus. And then the cilia, which are these tiny hair-like structures which are sweeping backwards and forwards, they actually end up moving the mucus up your trachea. And that's then when you get that sensation, like the tickly feeling in your chest that you need to cough. And that's what you're doing. You're coughing up the mucus to remove any mucus that might have dust or microbes to protect your lungs. The trachea then branch into two, which are the bronchi. So you have a right bronchus, is singular, bronchi is plural. So there's our right bronchus, and here is our left bronchus. So that's our two branching tubes from the trachea. They then transport the air into these smaller branches known as the bronchioles. And at the end of the bronchioles, you have these bunches of alveoli. So in alveoli, you have them in these bunches that looks like grapes, but a single alveoli or alveolus for singular. If we zoom in, this is what it looks like. And this is literally where gas exchange happens. So the alveoli is just an empty air sac. So this bit here in pale pink, that is the alveoli. And it's just made up of a single layer of cells making a wall. And because it's such a thin layer, it's just a single layer of cells. On the outside, this is a capillary, a blood vessel, and we've got red blood cells flowing in the blood around it. But because the alveoli is only made up of one layer of cells, that's a really short diffusion distance. So oxygen, if I just draw this on, oxygen from, let's not do yellow, let's pick a different colour. Let's go black. Oxygen, which would have been in that inhaled air, that's going to diffuse into the blood from the alveoli. And it's a really short diffusion distance. It only has to diffuse through a single layer of cells that makes the wall of the alveoli, a single layer of cells that makes the wall of the capillary, and then it is in. And same for the carbon dioxide that is diffusing out. I'll do the label here because I can't fit it in. It's got a really short diffusion distance. We're going to talk about some other advantages on the next slide, other adaptations. But the final thing we can see here in terms of adaptations, or structures present, I should say, is that the alveoli are surrounded by lots of capillaries. And as we zoom in, we can just see one capillary here, but you have lots of capillaries surrounding all of the alveoli. The alveoli is literally the site of where gas exchange happens. Gas exchange just means the diffusion of the gases. So oxygen diffusing into the blood and carbon dioxide diffusing out of the blood. So then if we have a look at the adaptations, you might want to screenshot this. So you've got this as a table to put into your revision notes. We've talked about the structures, and we said that the air travels from the mouth and nose through the trachea, through the bronchi, to the bronchioles, and then the alveoli, which are the air sacs where gas exchange takes place. Gas exchange is diffusion. So oxygen diffuses from the alveoli into the bloodstream. Carbon dioxide diffuses from the bloodstream into the alveoli. And because it's diffusion, we need to think about what are the key adaptations that we see to increase diffusion. One of them is having a large space, a large surface area. So you actually have millions of those alveoli in your lungs. So you'll have millions in this lung, millions on this lung as well. And because there are so many, that creates a massive surface area for this diffusion, this gas exchange to occur. We talked about the thin walls already. The rich blood supply, so this is where I said that the alveoli are covered in a network of capillaries. Now this is an advantage because it's a short diffusion distance, because the blood stream is right next to it, but also it helps to maintain a concentration gradient, because as soon as the oxygen diffuses into the blood, it's then transported away, and that then helps to maintain a concentration gradient. Finally is ventilation, which means inhaling and exhaling. And because we're constantly breathing in air, which will have a higher concentration of oxygen, and exhaling air, which has a higher concentration of carbon dioxide, this ventilation is maintaining the concentration gradient as well. So those are our key features, how they are maintaining or increasing the rate of gas exchange. So that takes us on to the blood vessels then, which are still part of the circulatory system. There's three main blood vessels that you need to know: arteries, veins, and capillaries. All three of them have a different structure and function. So let's have a look. One thing though, just to point out, as a way to help you to remember this, is looking at these two things. So arteries, I always think A, away. Arteries carry blood away from the heart. Veins have the word in them. They're carrying blood back into the heart or back to the heart. So that's how to remember which way round it is in terms of their functions. So here we have a picture of an artery. If it was sliced open and you were looking at the cross section, and the same for the vein and the capillary. And we've got our summary table here. So first of all, we've just said this: arteries is A, away, carry blood away. Veins is going in, so it's going towards the heart. And capillaries, that is what is connecting together, if I just go back here, the arteries to the veins, and it's where we get exchange of substances happening. So the thickness then, arteries have the thickest of the walls. Veins have a thinner wall, but the thinnest of all of them is a capillary, and that's because a capillary is just made up of a single layer of cells. The pressure of the blood is high in the arteries, it's lower in comparison in the vein, and it's at its lowest in the capillaries. Then if we look at the size of the lumen, which means the space in the middle where the blood flows through, the arteries have a narrow lumen. The veins have a much wider lumen. It's not always perfectly circular as well. And then the capillaries have the narrowest of them. And in fact, they're so narrow that you can only fit one red blood cell through at a time. So it really slows down the speed of the blood flowing through the capillaries because they're so narrow. Then if we think about valves, which we said were those flaps that prevent the backflow of the blood, you only get valves in veins. In terms of elastic fibres, we only have elastic fibres in the artery and veins, not in the capillaries. And you get mainly the elastic fibres in the artery, have a much thicker layer, and that's because they have to be able to stretch and recoil as the blood is pumped through them at high pressure. Finally, then we've got the functions. So the artery is transporting that oxygenated blood under high pressure to the rest of the body. The veins are transporting deoxygenated blood at lower pressure back into the heart. And the capillaries are the site of exchange. So for example, gas exchange at the alveoli. So a little bit more then on what a valve is. It's a thin, but it's still strong, flap-like structure that you have in veins, but also we looked at them in the heart. And they ensure blood flows in one direction only. So they prevent the backflow of blood. They also help to maintain the flow to the heart by closing and opening at the right time. That is going to make sure that the valves help to continue moving the blood to the heart. So they are really important in the heart and the veins overall to help prevent the backflow and to make sure blood is moving efficiently through the circulatory system. So then if we move on to the blood is an example of a tissue because it's groups of different cells working together to perform a particular function. And these are the four components of blood that you need to know. You need to know their appearance, adaptations that they have, and the function. That's a key thing, is the function. So if you want, you can screenshot this and then you can have that summary table in your notes. So the plasma, this is the liquid part of the blood. And if you actually were to separate it out, so you just had the plasma is this sort of yellowy straw-like looking liquid. So it's a watery liquid, and it contains all of the dissolved substances. Like it might have dissolved proteins, it might have hormones, there might be waste in there like carbon dioxide. So it's there to help to transport different nutrients and waste in the blood. The red blood cells are responsible for transporting oxygen. Now, although it says and carbon dioxide there, ignore that because that's actually something you don't need to know until A level. So you just need to know that they transport oxygen. They are biconcave, or in other words, disc-shaped, but biconcave is the phrase that they would want for the exam, which means they dip in on both sides. So it's concave on this side, but also on that side, so it's biconcave if you were to look at it from that angle. And that helps make it to be a little bit flexible, but also increases the surface area, so you get more oxygen diffusing in. We also have white blood cells. You learn more about this in the infectious disease topic, topic 3, but they are larger than red blood cells, irregular in shape, they're flexible, they can surround and engulf pathogens, but their main role is to defend the body against infection and disease. So it could be making antibodies, antitoxins, or engulfing pathogens. And then lastly, the platelets. You don't really need to know this information here about adaptations or appearance, but they are fragments of cells and they help to clot the blood. So that is what causes blood clots. If you have a cut, that's why you eventually stop bleeding. It's platelets trap all the red blood cells and help the blood to clot. Now, one thing it says in the specification that you need to be aware of is that if someone has lost a lot of blood, maybe due to a medical condition or a severe injury, they can have a blood transfusion, which is when you get someone else's blood transported into your body, basically a blood transfusion. But you need to be aware of, or be able to evaluate the risk of someone having a blood transfusion. And you aren't expected to know these reasons off by heart, but if they gave you this information, they'd want you to be able to say whether it's good or bad. And obviously the main good thing is it's going to replace the blood loss. So you'll be replacing the plasma, the red blood cells, the white blood cells, the platelets. The risk, though, could be you are at high risk of transmitting infectious disease, because if someone is giving their blood, maybe they had HIV or another virus and it wasn't detected, then you now have that virus in your body. Sometimes people have an allergic reaction. Sometimes people might reject the blood, meaning the immune system realizes it wasn't part of their body originally and it causes an immune response. But that's why they do blood type matching, to try and reduce the likelihood of that happening. And then sometimes iron overload, but I haven't actually seen this ever come up on the GCSE. So the top 3 are the main ones that might come up as an application question. So then we move on to what could go wrong. So coronary heart disease is an example of cardiovascular diseases. Cardiovascular means any disease linked to the heart or blood vessels. So it could be coronary heart disease or it could be heart failure. And you need to know a very little bit about this, mainly looking at how it can be treated. And it could be treated using certain drugs, mechanical devices, or even having transplants. So the drugs would be statins, mechanical devices would be things like stents, and then transplants could be a valve or it could be a heart transplant. Now you can screenshot this to have this as your summary, but I'm going to go through them all in more detail now. So coronary heart disease, this is if you have a buildup of this fatty material blocking your coronary arteries and therefore oxygenated blood and glucose aren't reaching the muscles, so they can't contract. We talked about that a little bit earlier. So what you can do then is, one option is using something called a stent, which are small tubes that get inserted into the arteries, those coronary arteries, and then you can either use something to inflate and push that tube outwards, or you can actually leave that metal mesh inside as well. And essentially what it is doing is pushing those fatty deposits backwards and making the artery a hollow tube again. So the advantages are that stents can often be inserted with very little surgery, and it can restore the blood flow, so it reduces your risk of a heart attack. The downside, though, is it is surgery, and all surgery comes with a potential risk of you getting infected. And you also might need to have this treatment repeatedly. So restenosis, you don't need to know that's a term. All it means is you might have to have that procedure again, which again puts you at higher risk of infection again. Then statins. So this is a drug treatment. So these are drugs that reduce cholesterol in your blood. And cholesterol is what makes those fatty deposits. So the advantage of this is it's not invasive. You don't have to have any surgery. You just take them as a tablet. They're preventative, so they can help reduce the progression of heart disease and the likelihood you might have a heart attack. But the downsides are they can have some side effects such as muscle pain, digestive problems, liver damage sometimes as well. And also you would have to take statins for the rest of your life to have that benefit continually. Then if we think about faulty heart valves, so heart valves can become faulty, meaning they either don't open or close properly, so you're not preventing the backflow of blood. So the treatments then are, you could have a valve replacement, and that could be a biological valve made from animal tissues such as pig or cow, or it could be mechanical. So that would mean that it's made from synthetic materials, which means it's not from a living thing. So it could be like a metal or some sort of carbon synthetic material. So the advantages are it can improve the heart function and reduce symptoms of valve disease. It is a long-term solution as well. Disadvantages though, because it would involve surgery, there's a risk of infection, and kind of contradicting that long-term solution is a long-term solution if you're going to have the mechanical in particular, but biological valves sometimes do wear out and you might need to have them replaced again, particularly if you had this procedure done when you were quite young. Next then is heart failure, and this occurs when the heart is unable to pump blood effectively, leading to poor oxygen supply to the organs and tissues. So you would either need a heart transplant or an artificial heart. So in terms of heart transplants, you are reliant on there being a donor. The advantages are it is going to restore normal heart function and therefore significantly improve life expectancy and quality of life. It can be a long-term solution. Often it might last for 10 to 20 years and you might need to have another transplant though. Disadvantages is the big one, availability of donors. There is a shortage of suitable heart donors, which means there might be a very long wait time. There's also a risk of rejection when you have any kind of transplant. So you'd have to wait for a donor that matches your tissue types and also take immunosuppressant drugs, which basically means it suppresses, so it lowers your immune response to try and stop it from rejecting the heart. An artificial heart is a mechanical device which can be used to keep a patient alive while they're waiting for a human heart transplant. So advantages are it is readily available and it can keep you alive long enough while you're waiting for a heart transplant. It is temporary though. It allows the heart to rest, so it is going to reduce the strain on the body, but it's not going to be like a lifelong solution. And that takes us here. So it's a limited lifespan. The artificial hearts aren't a permanent solution. Risk of complications as well as a risk of blood clotting. Now, for all of these treatments that we've gone through, it would likely come up as an application question where they give you more details and you have to pick out the pros and cons and write an evaluation. Next then is looking at health and disease, and our definition of health is the state of physical and mental well-being. So it's not just looking at the absence of disease, but it includes being free from any physical or mental health concerns. And there's two types of diseases we can consider: communicable, which basically means infectious, or non-communicable, which are the ones that are linked to lifestyle and environmental factors. And you've got some examples written here as well. So the relationship between health and disease is something that you need to be aware of. And the following factors can impact physical and mental health. So diet, stress, and life situations. So a poor diet and maybe lack of exercise and high stress as well, all of those can increase the likelihood of you developing a non-communicable disease such as heart disease. So that would be physical health. Stress and maybe life situations, we've got examples like if you became unemployed or poverty or there was some trauma, that can contribute to mental health conditions such as depression and anxiety. There's also some interaction between different diseases, meaning if you have one disease, it can increase your risk of developing a different disease. So for example, if we look at immune system defects, people who have a disease resulting in a weakened or impaired immune system, such as those with HIV, which then can develop into AIDS, are more susceptible to get infectious diseases because their immune system can't fight off the pathogens. Virus and cancer, there's a link here as well, because some viruses, like the human papillomavirus, or HPV for short, are linked to certain cancers, and HPV is linked to cervical cancer. Pathogens and allergies as well. So sometimes if you're infected with a pathogen, particularly in young people like infants and babies, it can trigger an allergic reaction, and that's when you then start to see skin rashes. And then physical illness and mental health. So if you have a severe physical illness or maybe a long-term health problem, that can negatively affect a person's mental health, and it could lead to depression, anxiety, or other psychological conditions as well. You also need to know the impact that non-communicable diseases can have. So, for example, cardiovascular disease or type 2 diabetes, lung cancer, liver disease, these are non-communicable diseases, meaning they're not infectious, but they can have big impacts, first of all, on an individual level. So a person who might have one of these diseases might have a reduced quality of life, chronic pain, chronic means it's ongoing. They might have limited mobility, so they can't move around as much, and emotional distress. In terms of the impact on a local community, if you have a lot of people, so a high prevalence of a non-communicable disease, that can put a strain on the healthcare system. So maybe in that local community, the GP surgery is really, really busy and there's too many people that need support, or it could link to maybe social care needs as well, or maybe in that local community there's just not enough people working because they're ill, and that's going to affect the economy. On a national level, governments face high healthcare costs for treatment, but also for prevention. It costs as well if there's a lack of workforce because they're too ill to work, and that can have an impact as well on the economy. And then a global level, non-communicable diseases are responsible for a significant portion of global mortality, so deaths, creating challenges in healthcare funding, public health initiatives to try and consider how to overcome them, and economic inequalities between countries as well. So here's just a summary then of some lifestyle factors which can increase the likelihood of you developing certain non-communicable diseases. So diets, we can see that there's a link here to high sugar, high fats, increasing the likelihood of obesity, cardiovascular disease, also diabetes type 2. Smoking is linked to lung disease and cancer. Alcohol, excessive alcohol consumption can lead to liver disease affecting liver function, brain function, and if you're drinking when you're pregnant, that can lead to fetal alcohol syndrome. Obesity linked to type 2 diabetes. Exercise, so if you are doing regular exercise, that actually reduces your risk of cardiovascular disease. Carcinogens, these are basically chemicals that increase the likelihood of mutations happening in your DNA. So if you're exposed to carcinogens, then that could increase the likelihood of you developing cancer. So carcinogens include things like in cigarette smoke, there's carcinogens. This here should really be split because radiation isn't a carcinogen, because carcinogen means chemical, but as a separate factor, being exposed to ionizing radiation also increases your risk of cancer. So all of this talk of risk then is looking at risk factors, and these are elements that increase the likelihood of you developing a disease. Doesn't mean you definitely will, just means you're more likely to. So here are some. So we've got life-based ones that we just looked at. Environment, so whether you're exposed to harmful substances and pollution or radiation. And a causal mechanism links risk factors to disease. So we do know that smoking can cause lung cancer. It's also a risk factor because it doesn't mean you will always get lung cancer, but it increases your risk. And we do know that the carcinogens in cigarette smoke can cause lung cancer. We also know being obese increases your risk, but being obese can actually result in diabetes, type 2 diabetes. And we know alcohol can cause liver damage. But again, it doesn't mean that it will always happen. It increases your risk. And we know that alcohol can cause these conditions, and the same with the other three. So the correlation doesn't prove it will always cause the disease, but we do know that there is that association between them. Next is looking at interactions. So many diseases result from interactions of multiple factors. So there's usually more than one cause. Cardiovascular disease, for example, might be because someone smoked, they had a high-fat diet, really stressful life, not exercising, and all of those together interacted to result in the cardiovascular disease. Cancer might be triggered by maybe your genetics partially, but also exposure to carcinogens or ionizing radiation and unhealthy lifestyle choices. And that leads us on to cancer. And you do need to know a little bit about cancer. So cancer is the result of changes in the cells. In particular, it's changes to the DNA in the cell, which results in uncontrolled cell division. So your cells just constantly divide. And because they are constantly dividing, it results in a tumour. And a tumour is a mass of abnormal cells, basically like a ball of cells that shouldn't be there. And it's because your cells are continually dividing, making more and more cells in a place you don't need those cells. And that uncontrolled cell division is because of mutations or changes in your DNA. There are two types of tumours that can form: a benign tumour and a malignant tumour. And here is our summary of the differences between the two. The main thing that you need to be aware of is that benign means it's not a cancerous tumour. Malignant means it is a cancerous tumour. And malignant or cancerous tumours can invade neighbouring tissues. So we can see here in a malignant tumour, shown in here, those red cancer cells, these can break apart from the tumour and then they might travel in the bloodstream and then lodge into another part of your body and cause another tumour, which is known as a secondary tumour. Benign tumours, though, are encased in this capsule. So they're stuck all together. It can only impact the exact location it's in, which means it makes it easier to try and remove. Whereas to try and remove a cancerous or malignant tumour is really difficult because the cells might have already spread to other parts. And it's really hard to know if you've definitely removed all the cancerous cells without removing some of the healthy body cells as well. So risk factors for cancer, some of these we've actually talked about already because it links to both lifestyle, but also there are some genetic factors. But here's just a short list. So smoking, diet, alcohol consumption, obesity, sun exposure. These all increase the risk of different types of cancers, which if you want, you can pause and have a read through. Then we've got the genetic risk factors. So some cancers, such as breast cancer, ovarian cancer, prostate cancer, there is a genetic link where you might have inherited some DNA, which means you're more likely to get a cancer than another person is who doesn't have that particular gene or that section of DNA. So understanding cancer in the context of risk factors then is the main thing is cancer is often caused by a combination of the risk factors rather than just one of those things we saw on the previous slide. So it could be a mixture of lifestyle and genetics or a combination of the different lifestyles. So for example, a person with a family history of breast cancer might have an increased risk of developing breast cancer because of their genetics, but if they maintain a healthy lifestyle, that helps to lower their overall risk. So lifestyle changes such as quitting smoking or improving diet can significantly lower the risk of many types of cancers, even if you had genetics that put you at higher risk. So the last bit of this topic then is looking at some plant tissues, organs and organ systems. So plant tissues, just as a quick reminder, a tissue is a group of cells with a similar structure and function. And plants have specialised tissues that work together to carry out essential functions such as photosynthesis, transport and growth. So here's a selection of the tissues that you need to know. Again, I've done it as a table so you can do a screenshot and then put it into your notes as well. So root hair cells, these are the cells that have got that long protruding part to provide a really large surface area, and it's where you have the absorption of water and mineral ions. The xylem tissue is the one that's the hollow tubes, and it's where you get the continuous column of water forming. Phloem tissue, you have those sieve plates, so it's hollow, but you have the sieve end plates in between, and this is where the sugars are transported. Meristem tissue is responsible for the plant growth by dividing, so that's where you have your stem cells. And then guard cells and stomata. The guard cells are on the lower side of a leaf, and they create this hole which is known as the stomata. Now, all of these are actually covered in more detail in my GCSE Topic 1 video, because this is where you learn about the specialised cells and also stem cells. So you go into this in more detail in the Topic 1 video. If you want to have a look at that, you can, or you can just read through this, screenshot it as well. So the leaf is a plant organ. And as a reminder, organs are groups of tissues that work together to perform a particular function. And the tissues inside of a leaf are the epidermis tissue, palisade mesophyll, spongy mesophyll, xylem and phloem, and the stomata and the guard cells. So here we can see that cross section through the leaf. So the epidermis, you have a top, so upper, and also a lower epidermis. That is often waxy and it's there to prevent water loss from the leaf. The palisade mesophyll is this layer here. We have lots of long oblong shaped cells with loads of chloroplasts in, and the chloroplasts are able to absorb light energy, which is needed for photosynthesis. The spongy mesophyll is this tissue layer here where you have lots of spaces where it's spaces for gases to diffuse into. So it helps with gas exchange in the leaf. The xylem and phloem, which we can't actually see in this image, but the xylem transport water and minerals, and the phloem transports the sugars. Stomata and guard cells we can see better here, because it's the guard cells that are controlling whether the stomata is open or closed, and the stomata are the holes. So we have two guard cells next to each other, and when they're turgid, which means they're hard, they're filled with water, they bend and curve, and that creates this opening, which is the stomata, where carbon dioxide can diffuse into and oxygen can diffuse out of. Water vapour also diffuses out, which isn't beneficial, but that's why the guard cells can control whether the stoma is open or shut. So a little bit more here. The stoma, or stomata for plural, are the small openings on the leaf, and that is where we have gas exchange, but also water vapour can evaporate. The guard cells are controlling the opening and closing of that stomata, and in bright light, guard cells are going to be absorbing water, and that makes them turgid or rigid, so they bend and curve and they open. And that then means plenty of carbon dioxide can diffuse in through the stomata and be used in photosynthesis. In dark or dry conditions, the guard cells lose water, so they become flaccid or unfirm, and that causes them to close together, and that closes the stomata, so you don't get water loss. Transpiration, this is the process of water movement through a plant, and it's basically evaporation from the leaves through the stomata. It's essential for transporting water and minerals through the plant, and actually it also helps to cool the plant. So here's the process. Water is absorbed by the root hair cells in the soil by osmosis. That water then moves into the xylem from the roots, and then it moves up the xylem as this continuous column of water through those hollow xylem tubes, and that is known as the transpiration stream. When the water reaches the stomata in the leaves, the water will then evaporate, and in the form of water vapour, it diffuses out through the stomata, and that's what we mean by transpiration. That loss of water at the stomata will then pull on that continuous column of water, so you get even more water being pulled up the xylem. So factors that can affect the rate of transpiration then, because it's basically evaporation, you've got to think of factors that affect evaporation. So that can be higher temperatures, because that means the water molecules will be moving faster because they've got more kinetic energy, and therefore evaporation happens faster. High humidity, that means you've got lots of water vapor in the air, and that reduces the concentration gradient, and therefore you have less evaporation and transpiration. Low humidity means you don't have much water vapor in the air, so you have a steeper concentration gradient and you get more transpiration. Air movement is linked to this idea. That should all be highlighted there. But if you've got lots of air movement, so it's really windy, that's going to blow away air that has lots of water vapor in it. And that way you're constantly maintaining a steep concentration gradient between the leaf and the air outside, and it increases transpiration. Light intensity will increase transpiration, and that's because at high light intensities it causes the stomata to open, and that means you've got a larger surface area for the water to evaporate out of. So that then takes us to the end. So it's time for you to test yourself. Pause this slide if you want to have a go. I'm not going to talk through the questions and answers. I'm literally just going to show them so that you can have a go. So here are the questions. Pause, screenshot, have a go. And here are the answers to those 10 questions. Again, pause, screenshot, and you can have a go at marking it. I hope you did find that helpful, particularly picking out all the key terms, key marking points and misconceptions. And if you did, that's exactly what my GCSE notes do. On each page, I go through the key terms, and there's examiner tips throughout where I show you the key marking points, misconceptions to avoid, and things that you should definitely say. So those are linked in the description below. That's it for this video though. I hope you found it helpful, and I'll see you in the next one.